HPS course file, and then those 3 remaining pictures on muscle contraction. So, if you saved 2 pages in the middle, great. Otherwise, you're probably going to need at least 2 pages, and then this is all the sliding filament theory, which is muscle contraction, but it's called the sliding filament theory. All of that goes together. And so, Haley, you may want to, so we did, the last thing that you probably have in there, is range of motion. Okay, so then skip 2 pages, and I sent you a video. If you can't find it, make sure you email me. So it'll have functions, and it'll have the beginning part of muscles. And then skip the next 2 pages, put the graphic organizer there and muscle fiber stuff, and then we're on the next 2. So this is the sliding filament theory. Muscle contractions are called the sliding filament theory because we're looking at that actin and myosin filaments and how they kind of interact with each other in order to contract a muscle. All right, so we looked at muscle fibers yesterday. That's basically the cell, the myocyte. I have mine on the right-hand side. And so that muscle tissue is made up of these fibers, so these muscle cells, especially when we're talking about skeletal muscle. So right now, we'll focus on skeletal muscle. They're these big, long, skinny fibers that make up, that are the cells. And these fibers can contract a maximum of about 50%. All right, so we looked at the sarcomere before. That is the segment of the myofibril that contracts. I have this highlighted green because I should have highlighted it yellow. No, I shouldn't. Wait. Oh, my mistake. It's on the next picture. What do I know? We didn't talk about the sarcomere before. Could have sworn we had. All right, sarcomere. Segment of that myofibril that contracts. Yep, this one we talked about striations. Maybe I didn't go back far enough. Oh, yeah, okay. I just didn't go back far enough. All right, so we did talk about the sarcomeres before. We talked about them in terms of the sarcomere is the unit that creates those perpendicular, vertical, if you will, stripes that are in striated muscle. So back then I said, it's going to make sense eventually. So here's that term that I was talking about. So on this picture right here, this one gives me a pretty good idea of what a sarcomere is. So this is a muscle fiber. So they're taking that muscle fiber, the skeletal muscle fiber, and they're taking that out of, and taking one of those myofibrils out of that muscle fiber so that we can take a look at it. When we look at that, here's a sarcomere, here's a sarcomere. That fiber, that myofibril, all the way down is going to have those designated regions, those sarcomeres. So that's where my green highlighting comes up. Yes, but I do have stuff underneath it. Hey, I just said not too long ago that notes are going to be boring today because Severin's not here. Yay! Okay, so the striation, the bands, are sacromeres. So this unit, so when we talked about bones, we had an osteon as a unit of compact bone. So we can think of the sarcomere as a unit of a myofibril, of a muscle fiber. So these guys right here have lighter and darker areas, and it kind of makes sense because it's a little bit lighter in these areas, the Z lines, because we don't have a whole lot of overlap of the fibers. We do have a darker area right here, a darker part of the band, because that's where the actin and myosin are overlapping with each other. So, it's more dense in those areas. The reason I liked this picture is because it gave me a good visual of the unit of the sarcomere. All right, so those fibers, the myofilaments. Actin and myosin. Actin is a thin protein filament. Lateral means outside of the median, to the side. Lateral means to the side. So actin is a thin protein filament anchored to the lateral sides of the sarcomere. So it's anchored to the Z line. Sarcomere, S-A-R-C-O-M-E-R-E. And before I go further, just want to point Myofilaments. So myocil means muscle. We did myofibrils before, fibers, filaments, that's even thinner, even smaller little piece, little part that we're talking about here. So actin is one myofilament. Myofilaments are what makes up the myofibril. Myosin is the other myofilament. This is the thick filament, thick protein filament, anchored to the medial, to the middle portion of the sarcomere. So since myosin is anchored in the middle, it's what basically pulls the actin into the medial area during that muscle fiber contraction. So on my diagram, I like this one a little bit better because it's a close-up. So this is just taken from here and kind of blown up just a little bit more. And I've added a little bit of labeling to my diagram, so I highlighted it as I had on my notes. But then on the sarcomere, I'm showing where my myosin, where my actin is, and this is my myofibril. These are my myofilaments of my myofibril. All right, so essentially it's zooming in. Unfortunately, it's not zooming in the same direction. So this is kind of zooming in, getting closer to see what you're seeing, to see what's going on. And so the same sort of thing is happening down here. It's essentially zooming in from the myofibril, getting closer to see what's happening. So here's my actin, my thin filament. Then when we come up here, this is our actin again. So notice in this actin diagram, it looks kind of like a little twisty fiber. And up here, here's the blue part is our little twisty fiber that represents actin. Tropomyosin is this strand, if you will, and troponin complex are these little cluster-looking things on the tropomyosin. On here, this is myofibril. Myosin, actin, those are the only words that I added. All right, so what are troponin, tropomyosin? Tropomyosin, that's that yellow line. It's a protein fiber that runs the length of an actin filament. It blocks the myosin binding sites on the actin. Tropomyosin is a protein fiber that runs the length of the actin filaments. It blocks the myosin binding site on the actin. The troponin, so this is not cat, Ca with a little plus sign, that's our calcium ions. Calcium binds to troponin protein, and it changes that troponin shape. Whenever calcium binds and changes the shape, it causes that tropomyosin to get moved off the myosin binding sites on the actin. That allows myosin to bind to it. You guys were good. All right, so looking at this picture right here, troponin complex, tropomyosin is the little fiber, actin is the blue twisty fiber. So when calcium is inputted into the system, when your brain says, hey, we need a muscle contraction, calcium is going to be released, and it's going to bond to these sites right here. When it bonds to these sites, you can even kind of see in this picture, we've got these little dots right there. Those would represent our myosin bonding sites. So when we looked at myosin down here, here's our little twisty actin fiber. See all these little spots that are coming out, these little projections, if you will, off the myosin? They're going to bind to those spots. So when they bind to those spots, they grab on, kind of like our fingers are grabbing on, and then it's going to pull so that the actin gets pulled closer to the center there. All right, so now let's look at those steps in a little bit more AP Bio detail. All right, a couple of additions I have to this. Oh, yes, and this was another really good visual about what's going on here. So let's look at the top part first. All right, the top of the picture, really nice visual. So this is before the input of calcium. This is after. Calcium is bonding to those sites, and notice how it moves the tropomyosin so that we have all of these little spots where the myosin can bond to. All right, so now, step number 1. And I would ask that you go ahead and highlight this as well. And you may want to make a note for yourself. ATP is hydrolyzed. What does that word mean?
Water.
Yes, what tells you water? Hydro is adding water, okay? What about the other word part? Break apart. So this is telling us that ATP is being broken apart. Hydrolyzed is being broken apart. So addition of water to break it apart. So up here you have ADP, which is adenosine diphosphate, and a phosphate group. ATP is hydrolyzed when the myosin head is unattached. Next step. ADP and P, in our phosphate group, are bound to the myosin as the myosin head attaches to the actin. So we've already had calcium input, we've already had calcium bond, we've had the tropomyosin move. So now all of those little doodads, these little dangly things, are finding spots to attach to that actin. ATP was hydrolyzed, creating ADP in our phosphate group. ADP and that phosphate group are bound to that myosin as the myosin head attaches to the actin. Next, and this stage right here is known as the power stroke. So we have ATP, it gets hydrolyzed, it's unattached. ADP and phosphate attached to myosin, myosin's attached to our actin. Then ADP and the phosphate group are released, and that causes that little myosin dangly thing to change its shape, and when it changes its shape, that's what's moving that actin filament. In order to reset, let's do this. There we go. In order to reset, then we take that ADP and phosphate group from the previous one, and then we bond them together, and it causes that myosin to return to its original position. Then we hydrolyze it. Then it attaches to the myosin, which attaches to the actin. Then that's released, causing it to move, which pulls the actin. Then we reform ATP. Whoops, it's kind of hard to do when looking at it. So, hydrolyzed hydrolysis, we're breaking down a polymer. So in this situation, we're breaking down ATP. ATP is hydrolyzed, ATP changes into ADP and the phosphate group. ADP could lose another phosphate group. Any idea what it will become if we break off another phosphate group? No. 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 It's going to lose another phosphate group. So ATP is adenosine triphosphate. How many phosphates? ADP, how many phosphates? ASP was a little bit closer, but we don't use S. I'm assuming you mean single, but we don't use single. What does you mean? Oh, makes sense, but no. So think to our polymer AMP. So what does the M stand for? You got it. Mono. So it would be AMP, adenosine monophosphate. And it is possible. Although they really don't like it. I mean, it takes a lot of effort, energy, whatever, to get it down to that point. Okay, sliding filament theory. The sliding filament theory are the steps for muscle contraction. That's what that means. That's what these notes are about. This is how muscles contract. We're going to find out when we get into the nervous system that potassium and sodium are really important components to creating that nerve impulse, to having that signal be passed on. When we looked back, way back at the beginning of PBS, we talked about rigor mortis. Do you remember what rigor mortis was? Right. And so then they don't relax. They stay contracted until things break down. So what's happening is that you have a release of that calcium that causes that contraction. But normally, after you're done with the contraction, there's processes in your body to remove that calcium, to flush it out, if you will, for lack of a better description, so that the muscle can relax. And technically, so it can relax, and so the muscle pair, remember they have to work in pairs, can allow that to get in its relaxed state, whatever that relaxed state is. However, when a person's deceased, it takes energy. It takes energy in order to relax and reset. And if a person's passed away, they don't have that energy. And so that's why this doesn't reset for them. If you have muscle cramps, eating bananas can help out because that potassium is going to help with those nerve signals and make sure the nerve signals are accurate. If you have muscle cramps, taking magnesium can help as well because magnesium can block calcium from binding. And so if calcium can't bind, it can't cause the contraction. Okie doke. All right, so muscular system. The only thing that you have left is you're going to add your graphic organizer, but that comes with the Build a Better Body situation. So everything else that we have in there. Okay, so let's talk just a minute about investigation number 1, because y'all are knocking things down like dominoes. Pew, pew, pew. Good job. So we've got HBS 141 done. Good. We've got arm muscle build done. Good. Notes for muscular system done. Good. So your investigation number 1, that's your AP biology. And in AP biology, you need to make sure that you have all of your data, have your graphs, but you're also going to add a CER. So your CER needs to address those scenarios that you have. The first scenario, first generation, 50th generation, that's essentially no pressures whatsoever, right? So that can kind of be your baseline. That can kind of be what you compare things to. Your second scenario, you have female preference. Your third scenario, you have predators. Your fourth scenario, you have female preference and predators. So what you really need to do when you're talking about these, and this isn't going to be, a one-sentence kind of situation. For this CER, you can write a claim. So your claim could answer that overarching question. We really don't need an E on this one. So if you want to put it on there, that's great, but you really don't need to. The R is what we're really looking at. I want you to analyze those graphs. I want you to analyze those situations and tell me what's going on. So situation number 2, look at it in terms of first generation and 50th generation. Do you see, is there a selection pressure that's affecting the number of spots? Compare it to the first and 50th generation. Compare it also to your first scenario, because the first scenario is like our control. So compare it to that as well. Do you see any kind of pressure? So that would be the first thing that you tell me. Yes, there is a selection pressure. There isn't a selection pressure. And then it's an explain. So tell me how you know that. What do you see? What are you looking at that shows there is some sort of selection pressure being applied to the male guppies in the number of spots? Or tell me what you see that tells you that there is no effect of female preference. Then for scenario number 3, that's just predators, no female, just predators. Look at that situation. Compare it first to 50th generation. Compare that situation to your first scenario, because your first scenario is like our control. Is there a selection pressure from the environment? Because predators would be like natural selection of pressure from the environment. Explain. How do you know that? Whether you say yes or no, what are you looking at? What are you seeing that tells you that? You can talk about first 50th generation. You can talk about comparing that to your first scenario. Then same thing for the last situation. Now the last one's a little bit trickier, so you do have to think about it a little bit more. Is there pressure there? Are there selection pressures at work here? Explain. So make sure you're telling me what you see to allow you to say that. Does that make sense? Is that it? Okay, give me thumbs down, it makes absolutely no sense. Thumbs up, it makes absolute perfect sense. Okay, so I see a lot of sideways, and that's fine. That's what I'm looking for, is I don't know how well you understand it. So what is the question? What would you ask me as far as writing that R part? Yes, go ahead. So you made 8 graphs, right? And each of your graphs should compare the number of spots on the male guppies in the first generation and the 50th. So far? So I want to know, was there a selection pressure that changed that population in terms of number of spots? Now keep in mind that first and 50th generation, let's say there's no pressure whatsoever. It's not going to be exactly the same. We're in nature. But when you look at the overall trend of your graph, generally speaking, is there a change? Does that help? Any other questions, especially if we had it sideways? So where are we at now? Are we still thumbs down, thumbs up, thumbs sideways? Are we still sideways? All right, so here's what I want you to do. You have the first scenario. Does everybody have all of their graphs done? Okay, so work on your graphs, and let me come around to the ones who's my sideways people. Okay, so let me come around, and I want to see, and I'll walk you through at least one of those as to like what I just said. And maybe having it in front of you and looking at it at the same time I say it might help out. So that reasoning part will be due on Monday. You can just drop off your course file in the tray, and I'll get that taken care of whenever I can. So far, we still have our makeup tours on Monday, so be prepared for that. It'll be the same way that we did that last time. We'll just have stations set up, and they'll run through, and we'll do that. It's McAllister, so in the morning, it's going to be the same, like 10 minutes at each section. Afternoon's a little bit different, but be prepared for that. I think other than that, so yeah, weather, stuff like that. So tomorrow, I'm still planning the test for tomorrow. Let's get that taken care of, knock that domino down as well. Should all go as planned. I mean, if we're out Thursday and Friday, they've already said that it will automatically be distance learning, and then they'll change their mind from that. So if we are not here tomorrow, then it's automatically distance learning, and they'll change things if they need to. And we'll go ahead, and those are the 2 things that we'll be responsible for Biomed. So we'll take the test on Canvas. It will be a timed test, so you will not have time to dig through notes, course file, to find answers. You need to make sure that you've gone over that stuff. Remember, there's plenty of things to study. There's a Kahoot. There's a couple of practice tests. There's a file on there that gives you some things, suggestions. And then the other thing would be is the 3rd quarter abstract. Work on the 3rd quarter abstract. heaven forbid, you should get it completely done and turn it in and not have to worry about that at all. Those would be the two things for tomorrow. Then we'll take Friday by ear and we'll see what happens. All right, questions? Okay, go ahead and work on those graphs for investigation.